So to answer these two questions, we can move on to the next slides. So after we make all the polymer, of course, it is not easy, right? We will have to drip them on the hot surface, and then by coincidence, by chance, then the monomer will link to each other. And it is not easy to form these polymer. Of course, we want to keep them. We want to keep the precious polymer. How do we do it? We form a pre-cell. We want to isolate all these biological molecules, all these macromolecules, all these polymer. And we can form a pre-cell by using fatty acid. So the pre-cell or the cell membrane is formed by the fatty acid, so that we can contain all these polymer inside the fatty acid membrane. So you can see that, okay, this is the dehydration process that we learned at the beginning of the semester. And you have a glycerol molecule, and then you have a fatty acid chain, and then one glycerol molecule can attach to 3 fatty acid chain. And then you have glycerol head and then 3 fatty acid tails. And this lipid, this kind of molecule, they are good enough. This fat molecule, they are good enough to form a membrane to contain all the polymer. After that, we want to develop a self-replicating molecule. Meaning that, okay, after we form a cell, we want to continue the continuation of this pre-cell. So when we want to continue with all these pre-cell, we need to have some kind of a molecule that can that will take care of this kind of process. So the first genetic molecule is not DNA; it's actually RNA. Well, you may think of why, but you know that because many people think that DNA is the one that contains the gene. You know, DNA is the one that contains the codon. DNA is the one that transcribes into RNA, right? DNA is the one that is transcribed into RNA. Then shouldn't it be DNA the first one? The truth is that it is the RNA. The reason is, look at the structure. You need to have double-stranded to form a DNA, but you only need single strand to form RNA. So that makes RNA easier to occur. So the first gene were actually the short strand of DNA. They can replicate themselves without any enzyme or protein, and they are basically using RNA as enzyme. So we call it ribozyme. So it's an RNA enzyme. That's why RNA is the primitive, the earlier form of the genetic material. So at first you have all the RNA monomer. Mono, that means they are all the nucleotide. And then they start to connect to each other to form the polymer of RNA, or we call it simple genes. And then they start, after they form simple gene, a single-stranded RNA, they can, based on the base pairing rule, they can form a complementary strand of RNA chain, so that they will become a double-stranded molecule. Then after that, they can start to have the replication process. One serves as a template, one serves as an original copy, one serves as a template to start the replication process. So do not think that, oh wow, we have pre-cell, then we have the self-replicating process. It takes a long time. It takes millions of years. And the changes is incremental. That means it is step by step, very slowly. It's not as fast as we go through it in the lecture. So that's it for this week's online lecture.